Okay, so the summary of the genetics, right? The principle of inheritance and variation. So, genetics is a branch of biology which deals with the principles of inheritance and its practices, right? Progeny resembling the progeny resembling the parents in morphological and physiological features has attracted the attention of many biologists. Mendel was the first to study these phenomena systematically, right? Mendel was the first, right? And while studying the pattern of inheritance in pea plants of contrasting characters, Mendel proposed the principle of inheritance. So, Mendel studied the pattern of inheritance in pea plants of contrasting characters. Now, Mendel proposed the principle of inheritance, which are today referred to as the Mendel's laws of inheritance, right? Now, he proposed that the factors that is later named as the genes regulating the characters are found in pairs known as alleles. So, the factors which was he named it the to the characters, right, regulating the characters are found in pairs and now known as the alleles. He observed that the expression of the characters in the offspring follow a definite pattern in different first on different first. Okay, so where it was like, okay, so he proposed that the factors, right, those are in pairs now known as alleles. He observed that the expression of the characters in the offspring follow definite pattern in different first generations f1 and second f2 and so on. So they follow the expression of the characters in the offspring follow definite pattern in different first generation and second, that is f2 and so on. So some characters are dominant over others, right? The dominant characters are expressed when the factors are heterozygous condition, law of dominance, right? It means like capital T L small t, in which the capital T is dominant, so it is tall. So the dominant characters are expressed when the factors are in heterozygous condition, law of dominance. The recessive characters are only expressed in homozygous condition. It means it can only express in the small t, not capital T small t, where capital T is predominant. In capital T, no t, no small t. So this is a recessive character that was now now you can see like why it's going downward, I don't know. Okay, so the residual characters are only expressed in homozygous conditions. The characters never blend in heterozygous condition. Now this is the law of segregation because the characters cannot be blend. Okay, a recessive character that was not expressed in heterozygous condition may be expressed again when it becomes homozygous, right? So if it is not expressed in the heterozygous, so it can be expressed in the homozygous. Hence the character segregate while the formation of gamete. This is the law of segregation, right? So Mendel's laws of inheritance, like first one is the law of segregation. No, first one is the law of dominance. Second one is the law of segregation, right? So not all characters show true dominance. So not all characters show true dominance, right? Some characters show incomplete and some show codominance, like so when Mendel studied the inheritance of two characters together, it was found that the factors independently assort and combine in all permutations and combinations. So this is the law of independent assortment, right? So when Mendel studied the inheritance of two characters together, so when he studied the two characters of together, it was found that the factors independently, they independently. So the factors, it means present, showing that regulating the characters, they show the independently assort, and combine in all permutations and combinations. That is the law of independent assortment. Now different combinations of gametes are theoretically represented in a square tubular form known as the Punnett square. So different combination of gametes, that is capital T small t capital T, if it's like we are talking about the two contrasting characters. So okay, so this is expressed in the Punnett square. Okay, F1 and F2 generation. Now the factors now known as gene on chromosome regulating the characters are called the genotype, and the physical expression of a character is called the phenotype. So the genetic composition is called the genotype, and the physical expression, that is a tall, dwarf, okay, pink flower, a white flower. So all these are the phenotypes. So it is the physical expression, right? Now after knowing that the genes are located on the chromosomes, A good correlation was drawn between the Mendel's law, that is segregation and assortment of chromosomes during meiosis. So after knowing that the genes are located on the chromosomes, right, a good correlation was drawn between Mendel's law, that is segregation and assortment of chromosomes during uh, during meiosis. So okay, segregation and assortment of chromosomes during meiosis, right. Now the Mendel's law were extended in the form of chromosomal theory of inheritance. So it was first Mendel's laws of inheritance. Now it is chromosomal theory of inheritance, right? So chromosomal theory of inheritance. So later it was found that the Mendel's law of independent assortment does not hold true for the genes that were located on the same chromosome because they showed the linkage. Now these genes were called as the linked genes, right? Now closely located genes assorted together, so they do not assorted independently. So they assort together and distantly, that is if they are placed at some distance, so distantly uh, so so the distantly located genes due to recombination, due to variation, due to recombination, that is exchange due to the crossing over during the meiosis one of the Pacchitin stage, right. So due to the recombination assorted, they assorted independently, so the linkage maps therefore corresponded to arrangement of genes on a chromosome, right. So this is the linkage maps, right. Now moving to the next paragraph, right. Now many genes were linked to sexes also and called as sex-linked genes. So many genes were linked to sexes also and called the sex-linked genes. Now the two sexes, male and female, were found to have a set of chromosomes which were common, another set which was different. The chromosomes which were different in two sexes were named as sex chromosomes. Okay, the remaining set was named as autosomes. So the remaining set was named as autosomes. Now in humans, a normal female has 22 pairs of autosomes and a pair of sex chromosomes, that is XX. A male has 22 pairs of autosomes same as the female and a pair of sex chromosomes as XY. Now in chicken eats matlab falls or birds. Okay, now sex chromosomes in male are ZZ and in females are ZW. Okay, so mutation is defined as change in genetic material. So mutation is defined as change in genetic material. A point mutation is the now now in here, like, a point mutation is a change of a single base pair in DNA. A point mutation is a change of a single base pair in DNA. Okay, now sickle cell anemia is caused due to the change of one base. So sickle cell anemia is caused due to change of one base in the gene coding for beta chain of hemoglobin, right. So GAG to GUG, that is glutamic to valine. So it is changed by glutamic acid to valine. Okay. Now inheritable mutations can be studied by generating a pedigree of a family. So inheritable mutations, that is inheritance of my inheritance which is given by the Mendel, and then chromosomal theory of inheritance. So all these, so if they have the mutations, can be studied by generating a pedigree of a family, right. Now some mutations involve changes in a whole set of chromosomes, that is polyploidy, or change in subset of chromosome number, that is aneuploidy. So some mutations involve changes in whole set of chromosomes, right, that is polyploidy. So it is n, so it gets 2n, it gets 3n, or change in a subset of chromosome number, that is aneuploidy. If it's like at the 21st chromosome, it gets 3 copies instead of getting 2 copies, like in the Down syndrome, right. So this is the case of the in aneuploidy. Okay, so this help in understanding the mutational basis of genetic disorders, right. Okay. Now Down syndrome is due to the trisomy of chromosome 21, where there is an extra copy of chromosome 21, and consequently the total number of chromosomes becomes 47. So it should be 46, but due to the extra copy of the chromosome 21, it gets 3 and the total becomes like 47. So in Turner syndrome, one X chromosome is missing and the sex chromosome is XO. So 40 to 44 autosomes are present, and if like 6 chromosomes should be XX, but it gets 1 is deleted, so XO. So this is rarely occur, so this is occurs in females, like this is the Turner syndrome. Now in Klinefelter syndrome, the condition is XXY. This can be easily studied by analysis of karyotypes. Okay, now the exercise. So this much is the exercise. So mention the advantages of selecting pea plant for experimenting by Needle, because it produces the offsprings in very little time, like maybe in a week, or the sexes are dimorphism, that is the sexes can be distinguished, right? And then it can be easily culturable. So so these are all the reasons given in the NCERT. So selecting pea plant for experiment by Mendel. Now differentiate between the following: dominance and recessive. So dominance is like one allele is dominant on the other, that it means it is selected. So capital T small t, so dominance is capital T, so it is tall. Then recessive, it is small t, so it is recessive. So homozygous and heterozygous. So homozygous, that is the same alleles, like capital T, and heterozygous, it has different allele, that is capital T and small t. Now monohybrid and dihybrid. Monohybrid means the crossing of the one contrasting character, and in dihybrid the two contrasting characters are two characters of contrasting pairs, right? They cross each other. A diploid organism is heterozygous for four loci. How many types of gametes can be produced? A diploid organism, that is NN and 2N, is heterozygous for four loci. Is heterozygous, that is capital T small t, for four loci. How many types of gametes can be produced? Explain the law of dominance using a monohybrid cross. So explain the law of dominance using monohybrid cross. Explain the law of dominance using monohybrid cross. So law of dominance, like monohybrid cross, law of dominance using a monohybrid cross. Okay, so you see the capital T small t being crossed, so the hybrid becomes like capital T small t, and then when you selfing it, then capital T small t, it becomes capital T small t, and small t. So this is the law of dominance using a monohybrid cross. Define and design a test cross. So test cross is used to identify the genotype of an unknown heterozygotes, like, okay, so and it is to know which genotype is this, we cross it with the recessive parent, recessive allele, like small t. Okay, using a Punnett square, work out the distribution of phenotypic features in the first filial generation after a cross between a homozygous female and a heterozygous male for a single locus. Using a Punnett square, work out the distribution of phenotypic features in the first filial generation without the distribution of phenotypic features in the first filial generation after a cross between a homozygous female and a heterozygous male for a single loci. Okay, so when a cross in made between tall plant with yellow seeds and tall plant with red seed, what proportions of the phenotype in the offspring could be accepted to be tall and green, dwarf and green? When a cross in made between tall plant with yellow seeds, so tall plants with yellow seed, capital T small t, capital Y small y, and tall plant with green seed, that is capital T small y, what proportions of phenotype in the offspring could be expected to be tall and green? So capital T small t, so what in the offspring could be expected to be tall and green? So like as you can see, like tall, like capital T small t, when you cross it like capital T small t with capital T small t, you will find like there is like 50%. Okay, so 50% is tall. And then what about the green? So when you cross smaller, you will 100% green. So what proportions of the phenotype in the offspring could be accepted to be tall and green? So tall 50% in here is 100%, so total 50% tall and green. And dwarf and green, I said tall and green and dwarf and green, I think it is 0%. Dwarf and green, I think it is 0%, right? No, it is not 0%. Dwarf small t, it is also 50%, I think. So two heterozygous parents are crossed. If the two loci are linked, what would be the distribution of phenotypic features in F1 generation for a dihybrid cross? So two heterozygous parents are crossed, that is capital T small t, capital T small t. Now if the two loci are linked, what would be the distribution of phenotypic features in F1 generation for a hybrid, for a dihybrid cross? Okay, so briefly mention the distribution of Th Morgan in genetics. So briefly mention the contribution of Th Morgan in genetics, right? So Th Morgan given the experimental verification of chromosomal theory of inheritance, but it was given by Sutton and Boveri, right? So this was given by Sutton and Boveri, right? And what is the pedigree analysis? So it is pedigree analysis is just like pedigree analysis is just like, I mean, so what is the pedigree analysis? What is pedigree analysis? Suggests how such an analysis can be useful. So pedigree analysis, it is kind of study of the ancestors and knowing the diseases and knowing the disorders. So this is the pedigree analysis with the help of the inheritance, and it can be pedigree also useful in inheritance, to knowing the inheritance pattern. So this is it. Suggests how such an analysis can be useful. How is sex determined in human beings? So it is determined by XX and XY. So this is determined by XX and XY. A child has blood group O if the father has blood group A and mother has blood group B. Work out the genotypes of the parents and the possible genotypes of the other offsprings. A child has blood group O if the father has blood group A and mother has blood group B. Work out the genotypes of the parents and the possible genotypes of the other offsprings. A child has blood group O if the father has blood group A and mother has blood group B. Work out the genotypes of the parents and the possible genotypes of other offsprings. So if the child has O, means IA, IVI, and yeah, it is, that's it. Like so other offsprings can also have the AB, and that's it. AB, A or B, and O. So that's it. Very it can be asked in exam. Okay, so explain the following terms with example, that is codominance and incomplete dominance. So codominance is means, in codominance, like what does it happen in codominance? Like codominance means uh, codominance means both the alleles express, right? Capital T small t. So so they both express themselves, like in blood groups like AB. So AB, when AB, so AB, so AB is the codominance. When IB they cross, then AB is the codominance. So this is how you can remember it. Okay, now codominance. So now incomplete dominance. Now in between, it is neither tall or neither drop. So it is like incomplete dominance. So in between them, like if you cross like violet flower and white flower, so you will get like the pink flower. So this is like incomplete dominance. It is not either white or not either red. No, not either violet or not either white. So what is the point mutation? So point mutation, it is like the change in a, so what is point mutation? Point mutation, it is a change in a genetic material. So this is so this is the point mutation, right? Now give one example: sickle cell anemia, a change in a single base pair. So this is the point mutation. Like GAG is converted to GUG, that is glutamic acid is replaced by the substituted by the valine. So this is how it does. Then who had proposed the chromosomal theory of inheritance? So it is proposed by the Sutton and Boveri, and experimental verification is given by the Th Morgan. So this is how it is. So mention any two autosomal genetic disorders with their symptoms. So mention any two autosomal genetic disorders with their symptoms. So the autosomal disorders, they have like the sickle cell anemia. Okay, sickle cell anemia, and the progeny is affected when both the parents will be carrier. So this is it. Like, so this is the two autosomal disorders, and another is the like another is the thalassemia. So these are all the autosomal recessive, but they are autosomal recessive, right? So they all are autosomal recessive. Okay, so they are all autosomal recessive. So this is how your chapter is done in here. Like, it's the summary part, and yeah, so this is it.